Welcome to Bio Bits and Bytes. Today we're diving into something that's, well, quietly becoming one of the biggest threats to global health. Imagine a world, and it's not far off, where simple infections become deadly again, where surgery, cancer treatment, things we rely on become incredibly risky. We're talking about antibiotic resistance.
It's definitely not science fiction anymore. It's a very real crisis happening right now. The sources we're looking at call it a critical threat, maybe one of the most vital threats the entire healthcare sector is facing.
And the numbers are just sobering. You shared data indicating, what was it, nearly 5 million deaths associated with resistance in 2019 alone?
That's right, 4.95 million associated deaths and 1.27 million deaths directly caused by bacterial AMR just in that 1 year.
Yeah.
And the impact is devastatingly unequal, hitting low and middle income countries the hardest.
It's like the miracle drugs, the antibiotics that revolutionized medicine, are starting to fail us.
Precisely. Bacteria are incredibly smart, evolutionarily speaking. They adapt. They're developing all these complex ways to survive the drugs we throw at them, even the antibiotics we've kept in reserve, the last line therapies.
Superbugs,
Yeah.
that's the term you hear. These are the bacteria that have learned to resist multiple different antibiotics.
Exactly. And the World Health Organization, the WHO, they're sounding major alarms. They've flagged certain bacteria as critical priority pathogens.
These are the ones we really need to worry about.
Yes, because resistance is already widespread, or the infections they cause are just exceptionally difficult to treat. Your sources list the big ones: CRE, CRAB, CRPA.
Okay, let's break those acronyms down a bit. CRE.
CRE is carbapenem-resistant enterobacterales. Carbapenems are a class of really important, powerful antibiotics.
Got it. And SHRAB.
Carbapenem-resistant Acinetobacter baumannii, another really tough gram-negative bug often found in hospitals.
And CRPA.
Carbapenem-resistant Pseudomonas aeruginosa, again, common in healthcare settings, notoriously difficult to treat.
Okay, so CRE, CRAB, CRPA. What else is on that critical list?
Well, there's the infamous MRSA, methicillin-resistant Staphylococcus aureus.
Heard of that one.
Also, Clostridioides difficile, or C. diff, which can cause severe diarrhea and is often resistant. Drug-resistant Neisseria gonorrhea. And ESBL-producing Enterobacteriaceae,
ESBL.
extended-spectrum beta-lactamase producers. Beta-lactamases are enzymes bacteria use to break down many common antibiotics like penicillin. ESBLs break down even more types.
So these aren't just minor threats. They're bacteria that can potentially shrug off almost everything we have.
In some cases, yes. Resistance to nearly all, or sometimes literally all, available antibiotics. That's the nightmare scenario.
So our mission today in this deep dive is to look at the science fighting back. We've got a stack of recent research papers and reviews you pulled together. We want to explore the new ideas, the novel compounds, the different strategies scientists are testing to try and outsmart these superbugs.
Right, we're asking, what makes these potential new weapons different? And crucially, why might they actually work when others are failing?
We've got some really interesting sources. There's one on a synthetic compound, COE2 hexyl, that looks really promising.
Yes, broad spectrum activity and, really excitingly, seems to evade resistance development in early tests.
And a review that really digs into how bacteria resist and looks at different approaches like targeting virulence.
Plus specific articles on other compounds like infuside for gram-positives, mention of cresomycin, and another on synthetic peptides designed to tackle biofilms.
Okay, so a good mix. Let's start by really hammering home the scale of this problem. We mentioned the death toll, the impact on LMICs, but it goes deeper, right? It threatens modern medicine itself.
Absolutely. It's not just about treating infections when they happen. So many medical advancements rely on effective antibiotics to be safe. Think complex surgeries, organ transplants, joint replacements.
Chemotherapy for cancer?
Exactly. All of those procedures carry an inherent risk of infection. We rely on antibiotics to prevent or treat those infections effectively. If our antibiotics don't work,
then those life-saving procedures become incredibly dangerous.
correct. The risk-benefit calculation changes dramatically.
And you mentioned something else that makes this even more complex: climate change. How does that fit in?
Yeah, one of the sources calls it a potential force multiplier. Changes in temperature and weather patterns can shift the geographical range of infectious diseases and their vectors, like insects. It can also affect how bacteria survive and spread in the environment. So you could see diseases, including resistant infections, spreading to new areas.
Adding another layer of complexity to an already huge problem.
Definitely.
Okay, let's go back to those critical pathogens, CRE, MRSA, etc. You said some can be resistant to all or nearly all antibiotics. Can you unpack what multi-drug resistant really means for us and why that all or nearly all is so critical?
Sure. So, multi-drug resistant, or MDR, is generally defined when a bacterium is resistant to at least one antibiotic in 3 or more different drug categories or classes. So it's not just resistant to one thing, it's built up defenses against several different types of attack.
Okay, so it's learned multiple tricks.
Exactly. Now, when we talk about resistance to all or nearly all available antibiotics, that's the extreme end. It means for a patient with that specific infection, doctors might have literally no effective treatment options left. Maybe there's one highly toxic drug they could try with severe side effects, or maybe nothing works.
That's terrifying. That's where medicine just stops.
It's the point where we effectively run out of options for that infection. It dramatically increases the risk of severe illness, long hospital stays, treatment failure, and death.
And compounding this, the pipeline for developing new antibiotics has slowed down. It seems like exactly when we need them most, we're not getting enough new ones. Why?
It's a major issue. There are significant hurdles. Scientifically, it's hard to find new ways to kill bacteria that they haven't seen before, but the economic challenges are huge too.
How so? Isn't there a massive need?
There's a massive need, yes, but the market doesn't always reward that need effectively. Developing any drug costs a fortune, billions sometimes. But for antibiotics, if you develop a great new one, the responsible thing to do is often to hold it back, reserve it as a drug of last resort to preserve its effectiveness.
Right, so you don't use it unless you absolutely have to slow down resistance.
Exactly. But from a business perspective, that means lower sales compared to, say, a cholesterol drug someone takes every day for years. Plus, there's always the risk that bacteria will develop resistance to even a brand new antibiotic relatively quickly, cutting short its useful lifespan and the return on that massive investment.
So less financial incentive compared to drugs for chronic diseases.
Pretty much. It makes antibiotic R&D a less attractive proposition for many pharmaceutical companies.
That makes sense, unfortunately. And the problem isn't just confined to hospitals anymore, is it? These MDR bugs are getting out into the community.
That's another really worrying trend highlighted in the sources. We used to associate these highly resistant bacteria primarily with nosocomial infections, those acquired in healthcare settings.
Okay, so nosocomial means hospital-acquired.
Correct. Hospitals, clinics, nursing homes. But now we're seeing these same MDR bacteria causing community-acquired infections much more frequently.
Meaning people are picking them up just out and about, not in a hospital.
Exactly. Outside of healthcare facilities. This is concerning because it means resistance isn't just a problem for hospitalized or vulnerable patients anymore. Common infections, maybe a UTI or a skin infection, things that used to be easy to treat at home, could now be caused by an MDR bug.
Making them much harder to treat for anyone, anywhere.
Right. It increases illness, deaths, healthcare costs across the board. It truly makes antibiotic resistance everyone's problem.
Okay, that really paints the picture of the threat. Let's shift now to how the bacteria are doing this. How do they actually fight back? What are their tricks?
Well, they are masters of adaptation. Their rapid reproduction and ability to share genetic information gives them a huge advantage. Resistance primarily comes about in 2 main ways. One is through spontaneous changes, mutations, in their own DNA. The other, very importantly, is by acquiring ready-made resistance genes from other bacteria.
Sharing the cheat codes, basically.
You could say that, yeah. Often through things called plasmids, which are small, mobile loops of DNA that can easily pass between bacterial cells, even between different species sometimes.
Okay, so they mutate or they share genes. Let's dig into the specific mechanisms the sources describe. What's a first line of defense for them, keeping the drug out?
That's a key one, especially for gram-negative bacteria. Remember, they have that extra outer membrane layer that gram-positives don't, making them trickier targets to begin with. This strategy is restricting entry. Think of it like controlling the gates. Gram-negative bacteria have protein channels in their outer membrane called porins. These let certain molecules in, including some antibiotics.
Okay, like little doorways.
Exactly. So bacteria can mutate the genes for these porins, maybe make the doorway smaller or oddly shaped, or simply produce fewer porin proteins overall. This makes the outer membrane less permeable to the antibiotic, making it harder for the drug to get inside to its target. The sources mention examples in Klebsiella, E. coli, Acinetobacter.
So tighter border control. But what if the antibiotic does sneak inside?
Then the bacteria might deploy efflux pumps. These are amazing little machines, really. They're proteins embedded in the bacterial membranes that act like tiny pumps. Their job is to recognize substances that look foreign or toxic, including many antibiotics, and actively pump them back out of the cell.
Like a bilge pump on a boat.
That's a good analogy, yeah.
Hmm.
If a bacterium starts making lots more of these pumps, or evolves more efficient ones, it can pump the drug out faster than it comes in. This keeps the internal concentration of the antibiotic too low to be effective. MDR efflux pumps are a big problem in bugs like Pseudomonas and E. coli.
Wow, okay. So block the entrance, pump out intruders. What else? Can they change what the antibiotic is
Mm.
aiming for?
Yes, that's another major strategy. Modification or defense of the antibiotic target. Most antibiotics work by binding very specifically to an essential molecule inside the bacterial cell, like a component of the ribosome needed for making proteins or an enzyme needed for building the cell wall.
So they interfere with some vital process.
Right. Bacteria can counter this by developing mutations in the gene that codes for that target molecule. The mutation slightly changes the shape or chemistry of the target. So the antibiotic can't bind to it properly anymore, or binds much less effectively. The drug is still there, but it can't lock onto its target to do its job.
Sneaky. Are there examples of this?
Plenty. The sources mention mutations in ribosomal RNA that cause resistance to drugs like linezolid K pneumonia and S. aureus. Another way is using enzymes to modify the target itself. For example, certain enzymes can add a chemical tag, like a methyl group, to the antibiotic's binding site on the ribosome, physically blocking the drug. Yeah, this happens with resistance to macrolides and similar antibiotics.
And MRSA fits in here too, right? You mentioned it earlier.
Absolutely. MRSA is the classic example of target modification. Its resistance comes primarily from acquiring a gene called MECA. This MECA gene tells the bacterium how to make a different version of a crucial cell wall building enzyme. These enzymes are called penicillin-binding proteins, or PBPs.
Okay, hold on. Can you quickly define penicillin binding proteins, PBPs, for us? And how do they relate to antibiotics like penicillin, the lectins?
Sure. PBPs are essential bacterial enzymes. Their main job is to build and maintain the peptidoglycan layer, which is the main structural component of the bacterial cell wall. They stitch together the building blocks. The laxam antibiotics, that's a huge class, including penicillin, cephalosporins, carbapenems. They work because they chemically resemble the molecules PBPs normally bind to. They trick the PBP, bind to it permanently, and basically shut it down.
So the bacteria can't build this wall properly.
Exactly. The wall weakens, and the cell eventually bursts. Now back to MRSA and its MECA gene. This gene produces a unique PBP called PBP2A. The key thing about PBP2A is that it has a very, very low affinity for most bolactam antibiotics. It just doesn't bind well to them.
Ah, so even if penicillin or methicillin is around, PBP2A can just ignore them and keep building the cell wall.
Pretty much. It allows the bacterium to continue synthesizing its cell wall even in the presence of drugs that would normally inhibit its regular PBPs. It's a bypass mechanism.
That's incredibly clever. So change the target? What about just destroying the antibiotic directly?
Yep, that's another major weapon. Antibiotic opposition via hydrolytic enzymes. Bacteria can produce enzymes that chemically attack and break down the antibiotic molecule itself. The most famous examples are the ublactamases, which we touched on with ESBLs. These enzymes specifically target and break the crucial blactam ring structure that's common to all penicillin-like antibiotics.
So they snip the active part of the drug.
Exactly. They hydrolyze it, cut it open, rendering the antibiotic completely inactive. There's a whole arsenal of these enzymes. Some, like penicillinase, are quite specific. Others, like cephalosporinases, target different lactams. Then you have the ESBLs with broader activity, and the most worrying are the carbapenemases, because they can destroy carbapenems, which are often our last resort against bacteria producing other lactamases. CRE and CCRAB make these.
Disabling our most powerful weapons. And there's another way they modify the drug, not destroying it, but changing it.
Right, modification of antibiotics through adding chemical groups. Instead of breaking the drug down, some bacteria have enzymes that attach small chemical tags like an azetyl group, a phosphate, or a nucleotide onto the antibiotic molecule. This modification changes the antibiotic's shape or properties so it can no longer bind to its target effectively, or sometimes it prevents the drug from even getting into the cell. The source mentions this happening with aminoglycoside antibiotics in bugs like Campylobacter.
Okay, wow. Block entry, pump out, change target, destroy drug, modify drug. That's a lot of defenses.
No,
But the sources also talk about biofilms. How do they add another layer?
Biofilm formation. This is a huge challenge clinically. Biofilms aren't just individual bacteria floating around. They are organized communities. Bacteria stick to a surface and then to each other, and secrete a protective slimy matrix around themselves made of sugars, proteins, DNA. A complex goo.
Like a shield.
Very much like a shield, yes.
Okay.
This matrix is a physical barrier that makes it really hard for antibiotics to penetrate deep into the community to reach all the bacteria within. But it's more than just a physical barrier. The environment inside the biofilm is different. Lower oxygen, different pH, fewer nutrients in some areas. This changes how the bacteria behave.
How so?
Well, many bacteria in a biofilm slow down their growth and metabolism. A lot of antibiotics work best against rapidly growing active cells. So these slower growing or dormant bacteria become much less susceptible. They can enter a sort of persister state, highly tolerant to antibiotics, even without specific resistance genes.
And you mentioned mixed communities.
Yes, and this is critical. Polymicrobial biofilms, where you have multiple different species living together, like P. aeruginosa and S. aureus, often found in chronic wounds or CF lungs, are particularly nasty. They can be even more resistant than single-species biofilms, possibly due to synergistic effects. Plus, living close together in this matrix makes it easier for them to swap those resistance genes we talked about earlier.
So biofilms are like fortified cities where bacteria are harder to kill and can even share weapons.
That's a pretty good analogy. They are incredibly difficult to eradicate once established.
Okay. Understanding these mechanisms makes it crystal clear why we need new approaches. Just tweaking old antibiotics might not cut it. So one idea mentioned is, instead of just trying to kill the bacteria, target their ability to cause disease, targeting virulence.
Exactly. That's a really promising alternative strategy. Traditional antibiotics go for the kill bactericidal or stop growth bacteriostatic. They target processes essential for the bacterium's basic survival.
But virulence targeting is different. Can you explain what virulence factors are exactly, and why targeting them might be a smarter approach in some ways?
Okay. Virulence factors are the specific tools or strategies a bacterium uses to actually cause disease in a host. Think of them as the bacterium's weapons or invasion gear. These include things that help them stick to tissues, invade cells, evade our immune system, produce toxins that damage tissues, move around, or build those protective biofilms we just discussed.
So not necessarily things they need to just live, but things they need to be harmful.
Precisely. Many virulence factors aren't absolutely essential for the bacterium's basic survival or growth, especially in a lab dish. They're specifically for interacting with and harming a host.
So why target those instead of essential life functions?
The thinking is about selective pressure. If you use a drug that targets an essential process, any bacterium that develops resistance has a massive survival advantage, and resistance spreads quickly. But if you target a virulence factor, say, you stop the bacterium from making a toxin or building a biofilm, you might disarm it. You make it less harmful, allowing the host's immune system to potentially clear the infection more easily without necessarily killing the bacterium outright.
So you're not creating that immediate life or death pressure that drives resistance so strongly.
That's the hypothesis. The selective pressure to develop resistance against an antivirulence drug might be weaker or evolve more slowly compared to a drug that directly kills. It's about neutralizing the threat rather than just annihilation.
That's a fascinating shift in strategy. What kinds of compounds are being looked at for this approach?
The review source mentions several classes. Chalcones, for example, have shown activity against biofilm formation and also quorum sensing.
Quorum sensing.
That's the communication system bacteria use. They release chemical signals to sense how many other bacteria are around them and coordinate group behaviors, like launching an attack or forming a biofilm, only when their numbers are high enough. Inhibiting quorum sensing can disrupt these coordinated actions.
Okay, interesting. What else?
Indoles and thiophene are also being explored as potential quorum sensing inhibitors.
Any specific examples showing promise?
Yeah, the review mentioned some thiazole-indolin 2,1 derivatives. These showed some direct antibacterial activity, but were also notably good at interfering with biofilm formation in S. aureus and P. aeruginosa. Another interesting one is a halogenated indole called 5-iodonolo. It seemed to mess with the development and movement of A. baumannii, and importantly, could disrupt or even get rid of established A. baumannii biofilms.
That's key. Disrupting existing biofilms is really hard.
It is. This compound also worked against E. coli and S. aureus biofilms, though apparently not P. aeruginosa. The source suggests it might work by generating reactive oxygen species, or ROS.
ROS, what are those again, and how would they help?
Reactive oxygen species, ROS, are highly reactive molecules containing oxygen, things like peroxides, superoxides. They're naturally produced in small amounts, but high levels cause oxidative stress, damaging cellular components like DNA, proteins, and especially cell membranes.
So the compound might be causing chemical damage to the bacteria, maybe hitting the membrane.
That seems to be the idea, yes.
Yeah.
Disrupting membrane integrity through oxidative damage. Other indole derivatives were also mentioned as being effective against skin biofilms caused by acne bacteria, C. acnes,
Yeah.
and S. aureus.
It seems like there's a lot of focus on disrupting those biofilms. The review also mentioned repurposing existing drugs, like antifungals.
Yes, that's another avenue. Looking at drugs already approved for other uses to see if they have unexpected antibacterial effects. The source mentions azole antifungals like itraconazole, fluconazole, clotrimazole have shown some activity against bacteria in lab tests. The review cautions that these might not necessarily be working via antivirulence mechanisms, but finding antibacterial activity in an already approved drug could potentially speed up development, since a lot of the safety testing is already done.
Makes sense. Okay, let's zoom in now on some of the specific new compounds highlighted in these sources. The first one, from that Science Open paper, COE22-hexyl, sounds particularly exciting.
It really does stand out. The first big finding was its incredibly broad-spectrum activity. The researchers tested it against 17 different clinical bacterial isolates.
And it worked against all of them.
Yes, all 17. And this wasn't just standard lab strains. It included a real mix of tough gram-negative CRE, crab, CRPA, E. coli, Klebsiella, gonorrhea, Shigella, Salmonella, Yersinia, and gram-positives like MRSA, MSSA, strep pneumonia. That's impressive breadth.
Especially hitting both gram-positives and gram-negatives effectively. But the activity against refractory strains seems even more important. Strains from actual patients failing treatment.
Exactly. That's where it gets really compelling. They tested it against bacteria isolated from sepsis patients whose infections weren't responding. One example was a CRE Klebsiella pneumonia strain that was resistant to, I think it was 20 out of 22 standard antibiotics tested clinically. 20.
Wow. Practically untreatable.
Pretty much. And against this highly resistant strain, COE22-hexyl showed potent activity. Its minimum inhibitory concentration, or MIC, that's the lowest concentration needed to stop growth, was only 4 gml. Compare that to ciprofloxacin, a common, powerful antibiotic, which needed a concentration of 128 gml against that same bug.
A huge difference. So it's potent even against the really tough ones. And it doesn't just stop them growing, it kills them.
Yes, the data indicates it has strong bactericidal power. It effectively killed Salmonella and MRSA in their tests, and did so relatively quickly, with kinetics similar to ciprofloxacin.
Okay, potent, broad spectrum kills bacteria. But the mechanism sounds like the really unique part. It hits multiple targets.
This is perhaps the most exciting aspect, and potentially key to why it might avoid resistance. It appears to have a multi-targeted mechanism, primarily focused on disrupting the bacterial cell membrane and its functions. Studies, particularly in E. coli, showed it messes with several critical processes all linked to the membrane. Septation.
Cell division, right?
Right, how the cell divides in two. It also impacted motility, how bacteria move, often using flagella anchored in the membrane. It interfered with ATP synthesis.
Energy production.
Yep, the cell's energy currency. It disrupted respiration, another core energy-generating process, often happening at the membrane. And it compromised membrane permeability.
Okay, let's clarify those. Septation stops reproduction. ATP synthesis and respiration cut off energy. And messing with membrane permeability, what does that do?
It means the membrane's integrity is damaged. It might become leaky, letting essential molecules out or letting harmful things in more easily. It disrupts the carefully controlled internal environment of the cell.
So it's heading cell division, movement, energy production, and the integrity of the outer layers all at once.
That seems to be the case. The hypothesis is that it does this not by targeting one specific enzyme, but by interfering with the complex interactions between proteins and lipids within the membrane, essentially gumming up the works for multiple essential membrane-associated processes simultaneously.
Why is hitting multiple targets like that so important?
Because it makes it much, much harder for the bacteria to evolve resistance. Think about it. If a drug has only one specific target, the bacterium might just need one lucky mutation in the gene for that target to become resistant. But if a drug effectively disables several different unrelated essential functions at the same time, the bacterium would likely need multiple independent mutations to occur simultaneously to counteract all those effects. That's statistically far less likely.
It's too much for the bacteria to fix all at once.
That's the idea. It overwhelms their ability to adapt quickly.
And this seems to be borne out by the resistance studies they did. The This is often the Achilles heel for new antibiotics.
Absolutely. And the results here were remarkable. They used rigorous methods to test resistance development, including standard serial passage over 21 days, and also a specialized device called a morbidostat.
Okay, what are serial dilution and morbidostat testing? Why are they important?
Serial dilution, or serial passage, is a common lab technique. You grow bacteria in broth with the antibiotic, then transfer a small amount to fresh broth with slightly more antibiotic, and repeat this over many generations, usually days or weeks. You measure how much the MIC increases over time. The morbidostat is like serial passage on steroids. It's an automated system that constantly monitors bacterial growth and uses pumps to precisely adjust the antibiotic concentration to keep the bacteria under continuous, strong pressure to evolve resistance. It forces evolution much faster and more aggressively.
So really tough tests for the bacteria. How did CoE2-hexyl do compared to a standard antibiotic like ciprofloxacin?
The difference was stark. In the morbidostat, ciprofloxacin resistance shot up rapidly. The MIC increased 64 to 128-fold. Bacteria quickly found ways around it. But with CoE22-hexyl, they found it did not evoke significant bacterial resistance under these intense conditions. In the 21-day serial passage, the MIC only went up 4-fold for MRSA and 8-fold for Salmonella. Even using hypermutable strains designed to mutate faster, resistance only increased 2 to 4-fold.
That's incredible. A tiny increase compared to ciprofloxacin.
It's a massive difference and strongly supports the idea that the multi-target mechanism makes it exceptionally difficult for bacteria to evolve high-level resistance quickly.
That's genuinely groundbreaking potential right there. Did they test it in animals too? Lab results are one thing, but in vivo.
Yes, they did crucial in vivo efficacy studies in mouse models of sepsis, using both the MRSA strain and that super-resistant CRE Klebsiella. The results were very positive. Mice treated with CoE22-hexyl had high survival rates, 9 out of 10 for MRSA sepsis, and a remarkable 10 out of 10 for the CRE sepsis. This compared to 0 survivors in the untreated control groups.
So it worked well in a living system against deadly infections. How did it compare to existing last resort drugs?
That's another key finding. They compared it to colistin, which is an older antibiotic often used as a last resort for CRE infections, despite known kidney toxicity risks. Against the CRE Klebsiella infection, CoE22-hexyl was significantly more effective than colistin. Survival was 10 out of 10 with CoE22-hexyl versus only 6 out of 10 with colistin.
Better than a current last resort option. What about safety in the mice?
The initial study suggested it did not appear intrinsically toxic in these mouse models. They didn't observe signs of major organ damage. Of course, much more safety testing would be needed for human use, but these early signs are encouraging.
Okay, CoE22-hexyl looks like a really strong contender. Broad spectrum, potent against resistant strains, multi-target mechanism, low resistance development in vitro, and effective with initial safety signals in vivo. That's a lot of checks in the boxes.
Yeah.
Let's move to the next compound mentioned, Infuside. This one sounds different.
Yes, Infuside, discussed in the ASM.org source, is another synthesized compound, but its focus seems narrower initially. It showed good activity primarily against problematic gram-positive MDR bacteria, specifically mentioning S. aureus, including MRSA and Enterococcus species.
Still major threats, especially in hospitals. What's special about Infuside?
The main point highlighted is that its mechanism appears to be distinct from other existing antimicrobials. The source doesn't detail the exact mechanism, but novelty is key here. A different way of killing bacteria means it might bypass the resistance mechanisms bacteria have already developed against current drugs.
A new angle of attack. How did it perform in tests?
In lab tests against bacterial colonies, it reportedly acted faster and more effectively than vancomycin, which is a workhorse drug for resistant gram-positive bugs. And in mouse models of skin infection with resistant S. aureus, Infuside treatments significantly reduced the bacterial load.
Any other interesting findings?
They also noted a synergistic effect when Infuside was combined with linezolid, another important drug for these types of infections. Synergy means the combination works better than you'd expect just by adding their individual effects. That could be clinically useful.
And what about making it? Is it practical?
The source mentions a potential advantage in its scalable synthesis. Apparently, the process developed doesn't rely on expensive or particularly hazardous solvents, which could make larger-scale manufacturing more feasible down the line.
Are there downsides or areas needing more work?
The main limitation mentioned is that, in these initial studies, it didn't show significant activity against gram-negative pathogens, so its spectrum is currently limited. However, the researchers are actively working on tweaking the molecule's structure to try and broaden its activity.
Okay, so infuside. Promising for tough gram-positives, novel mechanism, potential for synergy and easier manufacturing, but currently lacking gram-negative coverage. The Harvard Gazette source also briefly mentioned cresomycin.
Yes, cresomycin was highlighted as another synthetic compound showing potential. Specifically, it's shown activity against MDR, Klebsiella pneumonia, one of those critical CRE pathogens we talked about.
And it seems to have solid backing.
That's right. Its development emerged from research on component-based synthesis and has received support from key organizations focused on AMR, like the Blavatnik Biomedical Accelerator at Harvard and, importantly, CRBX.
CRBX, what's that?
CRBX is a major global nonprofit partnership. Its whole mission is to find and fund the most promising early-stage development of new antibiotics, vaccines, and diagnostics to combat drug-resistant bacteria. Getting CRBX funding is a significant vote of confidence.
So cresomycin is definitely one to watch, targeting a critical need. Okay, that covers the synthetic small molecules. Let's turn to the last candidate from the sources, peptide K6. This is structurally different, right? A peptide.
Correct. Peptide K6 from the PNAS paper is a synthetic peptide. Peptides are short chains of amino acids, smaller than proteins. This was very short, just 9 amino acids, and features tryptophan amino acids, noted W out. But the really clever part is its designed ability to self-assemble into nanostructured micelles in solution. They observed these structures were around 174 nanometers.
Okay, let's break that down. Peptide, short amino acid chain. Nanostructure equals tiny scale. Micelle is a kind of molecular cluster. How does forming these tiny balls help it fight bacteria?
Forming these nanostructured micelles offers potentially big advantages. Firstly, clustering together like this might protect the individual peptide molecules from being broken down quickly by enzymes in the body, potentially giving it a longer half-life. Secondly, and perhaps more importantly, when these micelles encounter a bacterial surface or a biofilm, they can deliver a very high local concentration of the peptide right where it's needed. It's like delivering a concentrated punch instead of just individual molecules floating around. This can significantly boost its effectiveness.
Concentrated delivery, that makes sense. And this peptide is aimed at particularly tough infections.
Yes, the source highlights its potency against P. aeruginosa and S. aureus, two really common and troublesome pathogens. But its real standout feature seems to be its effectiveness against polymicrobial infections, and especially biofilms formed when these two species live together.
The mixed biofilms we talked about earlier, the really hard ones.
Exactly. Those polymicrobial biofilms found in chronic wounds, on medical devices like catheters, or in the lungs of people with cystic fibrosis. These are notoriously persistent and resistant, partly because the two species can even enhance each other's resistance or virulence. Tackling these is a major unmet need.
So does peptide K6 just break up the biofilm or kill the bacteria inside too?
It seems to do both. The study reported potent antibiotic and antibiofilm activity. It killed free-floating planktonic bacteria, but also effectively tackled bacteria within established biofilms. And this was shown both in lab dishes, in vitro, and in a mouse model of catheter infection, in vivo.
How does it actually work? What's its mechanism?
Like COE22-hexyl. Peptide K6 appears to be membrane active. The evidence suggests it works by rapidly disrupting the physical integrity of the bacterial cell membrane, causing leakage and cell death relatively quickly.
Another membrane attacker. What about resistance development for this one?
Similar good news here, potentially. The study found that P. aeruginosa and S. aureus were less likely to develop resistance to peptide K6 compared to gentamicin, a conventional antibiotic used for these infections.
Why might that be?
The researchers suggest it might be related to efflux pumps again. If the peptide's main action is on the outer membrane or cell envelope, those internal efflux pumps might not be as effective at getting rid of it compared to drugs that need to get deep inside the cell. It's another potential way to sidestep common resistance mechanisms.
Okay, how about safety and getting it to where it needs to go in the body?
The initial in vivo safety studies in mice looked promising. When given by subcutaneous injection under the skin, it showed low toxicity with no obvious local reactions or signs of damage to major organs like the kidneys or liver. Given its effectiveness against lung pathogens like P. aeruginosa and S. aureus, and this apparent low toxicity, the paper suggests that aerosolized delivery, inhaling it directly into the lungs, like how inhaled gentamicin is sometimes used for CF patients, could be a really interesting future possibility.
Direct delivery to the site of infection in the lungs. That makes sense. So, summing up peptide K6.
It looks like a really promising candidate specifically tailored for those challenging polymicrobial biofilm infections. The combination of its unique self-assembling nanostructure, its rapid membrane-disrupting action, the lower propensity for resistance development seen in vitro, and the encouraging initial safety profile makes it a very interesting prospect.
It's genuinely encouraging to see such different approaches being explored across these sources. We've got broad-spectrum, multi-target molecules, specific gram-positive killers with novel mechanisms, peptides that self-assemble to butt biofilms. It's not just more of the same.
Absolutely. CoE22-hexyl, infuside cresomycin, peptide K6. They represent different strategies, different chemistries, different ways of thinking about how to overcome bacterial defenses. The virulence targeting approach is another great example of that innovative thinking.
And the research involves a huge network of scientists and institutions worldwide, right? This isn't just happening in one lab.
Definitely not. The sources list contributors from universities and research institutes across the U.S., UCSB, UC Davis, Sanford Burnham Prebys, Harvard Brigham and Women's, but also Singapore, Australia, India, France, Cyprus, China, Serbia. It's a truly global effort,
Yeah.
and it requires significant investment from funding bodies like the NIH, NSF, the Army Research Office, CARBX, and various international agencies. Combating AMR needs this kind of large-scale collaborative approach.
But even with these exciting leads, getting them from the lab bench to the patient's bedside is still a huge hurdle, isn't it?
Oh, absolutely. The path through clinical translation is long, incredibly expensive, and fraught with challenges. Many promising compounds fail in human trials for efficacy or safety reasons. There are regulatory hurdles, and those economic disincentives for antibiotic development we discussed earlier still exist.
So cautious optimism is needed.
Definitely. These are very promising findings, providing real hope, but there's still a long road ahead for any of these specific candidates to become approved medicines.
Still, it's a powerful counter-narrative to the doom and gloom around resistance. It shows that science is finding new ways to fight back.
Exactly. It demonstrates the ingenuity being applied to this problem. Novel mechanisms, resistance evasion strategies, biofilm disruption, virulence targeting. These offer genuine potential to regain ground against superbugs. They represent hope that we can develop therapies that bypass existing resistance and maybe even make it harder for new resistance to emerge.
Okay, so let's try to synthesize the key takeaways from our deep dive today. Antibiotic resistance is a profound, escalating global crisis, undermining healthcare worldwide. It's driven by the incredible adaptability of bacteria, who use a wide array of defense mechanisms, blocking entry, pumping drugs out, changing targets, destroying drugs, modifying them, and hiding in biofilms.
And the pipeline for traditional new antibiotics faces significant scientific and economic challenges.
But research is yielding exciting new strategies. We looked at targeting virulence factors to disarm bacteria rather than just kill them.
And we spotlighted several promising new synthetic compounds and peptides. CoE22-hexyl, with its broad spectrum, multi-target mechanism, and low resistance potential. Infuside, hitting tough gram-positives via a novel route. Cresomycin, targeting critical CRE. And peptide K6, designed to self-assemble and destroy difficult polymicrobial biofilms.
These represent diverse, innovative approaches that offer real hope in this ongoing battle.
They certainly do. It's a critical and dynamic field of research.
Which brings us to a final thought for you, our listeners. Bacteria have been evolving for billions of years. Adaptation is what they do best. This race for new antibiotics often feels like we're just trying to stay one step ahead. Will these truly novel strategies, the multi-target attacks, the anti-virulence approaches, the biofilm busters finally give us a more lasting advantage? Or is bacterial adaptation an inevitable force we'll always be chasing? And perhaps more fundamentally, what does this constant need for newer, more complex drugs tell us about our relationship with the microbial world and how we might need to rethink our use of antibiotics altogether? Some big questions to ponder. Thank you for joining us for this deep dive on BioBits and Bytes.